Essay: The causes and importance of variation and diversity in organisms. Variation in the genome of an organism may arise from random DNA mutation. Types of DNA mutation include substitution mutation. This is where one base is replaced by a different base. Inversion is another type of mutation, where a section may be more than one gene involved that has been inverted. Addition. This is the addition of a base pair into the DNA base sequence. This can lead to a frame shift in the DNA, such that when the mRNA is made from the DNA and it is read by the ribosome, the sequence of bases for each amino acid after the mutation may result in a change in the amino acid coded for. A deletion mutation is similar, in that the loss of a base can result in a frame shift. A frame shift can result in a long sequence of amino acids being changed, or may lead to truncation, i.e. cutting short of the mRNA or amino acid sequence, leading to a shortened non-functional protein. Such mutation in the DNA can have an effect on the organism because of the resultant change to the protein. In the case of non-functional protein, an organism may not be able to survive and may not reproduce and pass on their alleles to the next generation. In the case of cystic fibrosis in humans, those with cystic fibrosis mutated protein cannot release chloride ions into the air spaces in the lungs, and this produces a thick, sticky mucus that can harbour bacteria and lead to lung infections. Loss of lung function greatly reduces life expectancy, and a person with cystic fibrosis can die from pneumonia before they reach an age that they are able to reproduce and thus pass on alleles. This would reduce the number of alleles in the population. However, it is possible for heterozygotes to survive quite well, and this maintains some cystic fibrosis alleles in the population. In contrast, there are examples where mutations result in proteins that have improved function. In bacteria, a mutation in the plasmid DNA can lead to the development of a protein able to prevent death of the bacterium from a particular antibiotic. This is an example of antibiotic resistance. Genes for antibiotic resistance can be passed between bacteria and do not have to pass from parent to daughter cell by binary fission. Evolution of a population of antibiotic-resistant bacteria can occur when the bacterial population is exposed to the antibiotic. Only those containing the antibiotic resistance gene will survive, and this will lead to the eventual rise in the proportion of the population that are resistant to the antibiotic. This is advantageous to the population of one species of bacteria, and disadvantageous to the population of bacteria that is not resistant to the antibiotic. This is an example of interspecific competition. Other animal species can show interspecific competition, and this can lead to the co-evolution of traits. For example, in eagles, which are a bird of prey, their hemoglobin protein has a lower affinity for oxygen. This results in more oxygen being delivered to the tissues and thus allowing muscle tissues to aerobically respire and deliver more ATP for more muscle contraction, for more successful speed and therefore capture of prey when in pursuit. The population of prey species may in turn change the proportion of a particular trait in the population because of the eagle acting as a selection pressure. This would select a particular type of genetic variation that was advantageous for the prey species. This kind of natural selection is called directional selection. Similar examples have been observed with the evolution of the horse, which became taller and faster over millions of years due to the variation in the population created by random genetic mutations resulting in slight changes in the proteins which were advantageous to the individual. In this case, the selection pressure would have been the predator chasing after the horse. Aside from interspecific competition that leads to the selection of a particular trait that shows variation, there are also examples of intraspecific competition that lead to the selection of particular traits within a species population. For example, with cows, males are bred for their muscle mass. Farmers have used natural variation within the population and artificial selection leading to the selection of more muscly males being bred and muscly females to produce offspring that will yield more meat. However, due to the nature of the organism, there are issues with becoming too muscly. If the individual offspring is too large at birth, it is possible that the result of birth will lead to death of both the offspring and the mother, thus removing these alleles from the population. If the offspring is too small, they may not survive or may not mature. This is a type of stabilizing selection. This is when most of the offspring born to cows have a similar birth weight because those at either end of the spectrum in terms of their variation in birth weight, do not survive into adulthood and do not pass on their alleles to the next generation. This reduces the variation within the gene pool, which is advantageous for the genes concerned with childbirth as it ensures that the offspring will live and survive and so do their parents, but other alleles that may have been found with the disadvantageous alleles for childbirth might also be lost. A reduction in the gene pool is disadvantageous to the population because it means that if a pathogen infects the population, then due to the lack of genetic variation within the population, it is less likely that any individual will contain the alleles needed to survive the pathogen. In this case, we would say the species is critically endangered, as it is just a matter of time before the whole population is made extinct. Changes in the population due to variation can occur in separation within the same species. This is an example of geographical isolation, and reproductive isolation can occur, leading to disruptive selection. In the case of the Svalbard reindeer, a single population of the same species found itself geographically isolated due to the combination of genetic variation within each population and the differences in the selection pressures experienced by each population. In the two separate environments, the two populations evolved separately, one becoming hardier to the harsher climate and lower nutrient availability, and ultimately this led to the evolution of separate species, eventually becoming reproductively isolated and unable to interbreed to produce fertile offspring, which is the definition for species. This example of where variation in the population can lead to the evolution of many species from one original species was observed by Darwin and Wallace in separate parts of the world. Darwin documented the evolution of the finch species on the different islands of the Galapagos archipelago. This example of disruptive selection highlights the importance of variation within a population to enable populations to adapt to differences in their food sources and habitats such that they are more likely to survive and pass on the alleles. The evolution of species as outlined by Darwin has allowed the principle of evolution to be explored. With new DNA technologies and the ability to sequence the genome, it is possible to find the differences in the base sequence of groups of organisms and from there determine how recently they shared a common ancestor. The more similar the base sequences in the DNA, the more recently they shared a common ancestor and the more closely related the two species will be. The variation observed by Darwin may arise from random genetic mutations leading to a change of the base sequence, but there are other sources of variation in the new combinations of alleles. This can arise during meiosis. In meiosis 1, association of bivalents and the exchanging of genetic material at a chiasma leads to new combinations of alleles in the sister chromatid. In addition, following prophase 1 and the chiasmatic formation in meiosis 1, metaphase 1, the homologous pairs of chromosomes line up on the equator by random segregation. This leads to the random segregation and in turn leads to new combinations of alleles of the bivalent pairs. Similarly, in meiosis 2, metaphase 2, there is more random segregation, as it matters which way round the sister chromatids line up when the chromosomes line up on the equator. These new combinations of alleles can lead to further variation during fertilization when it is completely random which sperm will fertilize the egg cell. All of this leads to greater variation within the population of a species and hence the rise of sexual reproduction and meiosis as a means to increase genetic variability. In some species there is only asexual reproduction, and this can reduce the genetic variation since all daughter cells will be genetically identical to the parent cell, and this makes populations susceptible to the same drugs and diseases and more likely to become extinct. Combinations of alleles give only the story of variation within the genes that code for proteins, but the exons that code for amino acids between the exons that code for amino acids to make a protein are introns. The introns can have a wide range of base sequences as they do not code for amino acids and do not join together to form proteins, and it is these genetic variations that are exploited as a part of genetic fingerprint and using DNA to identify people at a crime scene or parentage. For example, if the same enzyme is used to cut up individual A, B and C's DNA, then that contains both introns and exons. Since most people have very similar exons, by cutting in the introns, it will be possible to get differences between individuals A, B and C in terms of the lengths of DNA fragments that are produced by the same enzyme. These fragments can then be separated using gel electrophoresis, and this allows individuals to be identified. It allows clearly to see when someone has been at a crime scene and exclude people who have not been there, or at least did not leave any DNA evidence there. Gene technology has utilized the knowledge of genes as the cause of variation and the advantageous nature of these variations to enable genes from one individual to be taken and placed into another organism so that the second organism can make the same proteins found in the first organism. An example of this is the sweet potato. Some types of sweet potato do not contain very much beta-carotene. By isolating the gene for beta-carotene and inserting it into sweet potatoes that do not carry much, populations of humans that suffer from night blindness due to a lack of vitamin A and beta-carotene can now grow their own source of beta-carotene in the form of sweet potatoes, in this way artificially increasing the genetic variation within the plant population and helping to improve the population survival of humans.